Welcome everyone to lecture on cells. We're going to talk today about identifying different cell types. By the time we're done with this lecture, you should be able to demonstrate an understanding of basic cellular structures and functions, identifying different cell types, and here I'm talking about prokaryotic versus eukaryotic cells and plant and animal cells. Compare cellular structures between cell types and describe the functions of those cellular structures. Now, there are two major types of cells. Prokaryotic cells, these are from the domain bacteria and archaea. Bacteria, I think we're all familiar with. Archaeans are another group of organisms. They're also microscopic. They share a lot in common with the cellular structure of bacteria, but we do know that they're genetically distinct enough from bacteria to classify in their own domain. We'll talk more about classification later this semester, but if you remember way back to high school biology, you talked about domain, kingdom, phylum, class, order, family, genus, species. Domain is the largest category we have for classifying living things. There are only three domains: domain bacteria, domain archaea, and domain eukaryota. So prokaryotes include domain bacteria and domain archaea. Archaea are microscopic, kind of extremophiles. You might find them in like hot springs, or they actually also live in the rumen of ruminants. Excuse me. And they produce methane. I don't know if you've ever heard that cows produce a lot of methane because they're ruminants. And they do that because they have these archaeans that live in their guts that actually produce the methane. So the first type of cells are prokaryotic cells. The second type of cells are eukaryotes from domain eukarya, and that includes protists, fungi, plants, and animals. One thing I do want to point out real quick while we have this slide up is the difference in size. So animal cells are about 10 micrometers. Well, that's the size here. So they're probably on the order of, like, these cells are maybe 30 micrometers long. And remember, there's 1,000 micrometers in 1 millimeter, and 10 millimeters in a centimeter. So they're very tiny, they're microscopic, but they're much bigger than these prokaryotic cells. So on this one, we see the scale bar here is about half a micrometer. So all told, this cell is maybe 3.5 micrometers long, about 1/10 the size of the eukaryotic cells. Excuse me. All right, so a prokaryotic cell, or in this case specifically a bacterial cell, we see the same scale bar here, half a micron, and about 3 micrometers long. Some bacterial cells have structures on the outside, like fimbriae or flagella, to help them move. They lack a nucleus, so instead of having a membrane-bound nucleus, their genetic material is clustered together in the center of the cell, and we call that clump of DNA a nucleoid. Bacteria also have a single circular chromosome, which is another difference from eukaryotic cells. They do have ribosomes, but the ribosomes in prokaryotic cells are different than the ribosomes in eukaryotic cells. They have a plasma membrane and a cell wall that helps give them rigid structure. So let's compare prokaryotic to eukaryotic cells. Prokaryote, the prefix pro means before. Karyote means kernel, so before kernel. And you can kind of think of a cell as like a kernel of life, right? The prefix eu means true. So eukaryotic would be a true kernel. Prokaryotic cells have no nucleus. They just have that clump of DNA in the middle called a nucleoid. Eukaryotic cells, however, have a nucleus with a membrane, the nuclear envelope. Prokaryotic cells are filled with cytosol or cytoplasm. So are eukaryotic cells. There are no organelles in a prokaryotic cell other than ribosomes. Eukaryotic cells have membrane-bound organelles with specialized structure and function. Prokaryotes are small in size. They're about 1/10 the size of a eukaryotic cell. And we tend to think of prokaryotic cells as being evolutionarily primitive. We think of early cells when life first evolved were probably most similar to prokaryotic cells. Eukaryotic cells are more complex. Prokaryotic cells come from domains bacteria and archaea, and eukaryotic cells would be from things like plants or animals. Here's a good scale to compare size. So, we measure human height in meters. The length of some of your nerve cells could also be close to a meter in length. A chicken egg, you'd probably measure in centimeters. A frog egg in millimeters. A human egg is probably something you'd measure in hundreds of micrometers. Most plant and animal cells are somewhere between 10 and 100 micrometers in length. The nucleus and most bacteria and mitochondria, these are things that are from about 1 to 10 microns in size. Micron and micrometer, by the way, are two different words for the same unit. They're synonyms. All right, the very smallest bacteria we know about are just a little bit over 100 nanometers in length. Viruses are smaller still, and ribosomes below that. Proteins and lipids are in the single digits of nanometers. Small molecules and atoms might only be 1/10 of a nanometer or more. Now, these arrows here also show us what we can see, what we have to use to be able to visualize and see these things. Now, you can see things that are, maybe from 1/2 millimeter and up. You can see unaided with your eyes. We like to use light microscopes when we're looking at things from a few millimeters down to micrometer scale. You can use the light microscope. And then to see anything smaller than a few microns, you have to use an electron microscope, which is a very powerful type of microscopy that allows you to see things that are down to the scale of a few nanometers. All right, so next we're going to categorize structural and functional similarities in cells. First, we'll start with the nucleus. When we're talking about structures here, if it's a membrane-bound organelle, it's something that we'd only find in a eukaryotic cell, right? All right, so a nucleus has a membrane. It's only found in eukaryotic cells. It functions as the control center of the cell. It contains DNA and mRNA. It's surrounded by a double membrane, or the nuclear envelope. It's continuous with the rough endoplasmic reticulum. And it has holes in it, nuclear pores, that control what enters and leaves the nucleus. And if you look, here's an electron microscope image of the pores, the holes on the surface of the nuclear membrane. These are what let things in and out of the membrane, or in or out of the nucleus. The nucleus also contains chromatin, a complex of DNA and proteins that makes up the chromosomes. Most of the time, when a eukaryotic cell is just hanging out being a cell, the DNA is in the structure of chromatin. It's kind of unraveled, and I kind of picture it like a bowl of ramen noodles. It's just loose so that the DNA can actually be used to make proteins. All right, there's also a nucleolus. This is the region where ribosomal subunits, rRNA, and proteins are formed. And it kind of looks like a spot on the nucleus. All right, here's a good picture of the nucleus. So if this structure here in the center that's purple is the nucleus, we see that the membrane of the nucleus flows into the membrane of the rough endoplasmic reticulum. It's basically the same membrane that continues on, right? You can see the squiggly chromatin here spread out within the nucleus. And then there's a dark spot on the nucleus called the nucleolus. These little sea urchin looking structures, these are the nuclear pores. And there are little holes that can open up to allow mRNA to exit the nucleus. All right, next up we have ribosomes. The function of ribosomes, their job is to make proteins, protein synthesis. They're made up of rRNA and protein. And they're always built of two parts. There's a large subunit and a small subunit, which you can see in the model here. Large subunit, small subunit, and that makes a ribosome. There are free ribosomes that float around in the cytosol. These produce proteins that are used within the cell. There are also ribosomes that are bound, they're stuck to the endoplasmic reticulum, and they make proteins for export outside of the cell. The endomembrane system is a structure that regulates protein traffic and performs metabolic functions. The endomembrane system consists of, so we've got the nucleus up here, which is surrounded by a double membrane. It flows into the rough endoplasmic reticulum, and we call it rough because it's studded with ribosomes and it looks bumpy on a microscope. There's also smooth endoplasmic reticulum. Same membrane, but it lacks the ribosomes. Then there's usually a space filled with cytosol, and in this side of that void, there is the Golgi apparatus. More membrane, right? It's kind of like layers and layers of membrane. Kind of like if you were to stack up a bunch of whoopee cushions or something. But layers and layers of membrane here. And what happens is when proteins are produced in the rough endoplasmic reticulum, they get passed along through the spaces in the membrane until they reach the outer edge. Then they're packaged in a piece of membrane from the endoplasmic reticulum, and they travel across the cytoplasm space to reach the Golgi apparatus. There, the membrane is absorbed by the Golgi, and the contents of that little packet flow through the voids in the Golgi apparatus to get to the other side, where they bud off in another little bubble of Golgi membrane. That bubble reaches the surface of the cell membrane. The membrane gets absorbed by the cell membrane, and the contents that were in that little packet are expelled from the cell. So the endomembrane system, nuclear membrane, endoplasmic reticulum, and Golgi apparatus are important for trafficking and packaging products or proteins produced by the cell that are needed elsewhere. All right, so the endoplasmic reticulum, it's a network of membranes and sacs made of membrane. There's two types. There's the rough ER with ribosomes on the surface, and the function of this rough ER is to package proteins for secretion, send transport vesicles to Golgi apparatus, and make replacement membrane. The smooth endoplasmic reticulum has no ribosomes. It functions to synthesize lipids, metabolize carbohydrates, detoxify drugs and poisons, and store calcium ions. All right, here's a picture of endoplasmic reticulum. Here you can see an electron microscope image showing the smooth endoplasmic reticulum here and a rough endoplasmic reticulum. You can see all the bumpy little ribosomes here. All right, the Golgi apparatus. The function of this is to synthesize or to make and package materials, small molecules for transport in vesicles. Those are those bubbles of membrane. And the Golgi also produces lysosomes. The Golgi looks like a series of flattened membrane stacks called cisternae. I've heard people describe this also as looking like a stack of pancakes. It has a cis face that receives the vesicles from the rough endoplasmic reticulum, and it also has a trans face on the outside, so facing the cell membrane, and that's where vesicles are shipped from. I mentioned lysosomes. Lysosomes are for intracellular digestion. They recycle cells' materials, and they're also responsible for programmed cell death, or apoptosis. They contain hydrolytic enzymes. All right, so if we look at the images on the left, you can see the little lysosome down here, the nucleus. All right, and then this is another electron microscope image. And this one has a vesicle containing two damaged organelles. There's a mitochondria fragment and a peroxisome fragment. And what's going to happen here is the lysosome will attach to that mitochondrion and break it down so that the building blocks, basically, that made up the mitochondria can be used to make something else the cell needs. Vacuoles are a storage organelle. They store materials like food, water, minerals, pigments, and even poisons. They're membrane-bound vesicles like the others we've talked about. And an example of this would be like a food vacuole or a contractile vacuole. Plant's large central vacuole, and this is usually a good clue that you're looking at a plant cell, is usually more than half of the space, but not always, inside a plant cell is occupied by the central vacuole. Membrane-bound vesicles. Okay, plants use these large central vacuoles to store water and ions. Ions are electrically charged particles, but they're typically things like salt or sugars, things that dissolve easily. All right. So here's a close-up again of the nucleus, the endoplasmic reticulum. There's rough ER and some smooth ER in there. Here's the vesicle leaving the Golgi and leaving the endoplasmic reticulum and heading to the Golgi. And then here's a trans Golgi leaving the Golgi apparatus behind and heading to the cell membrane for export. Okay, here's a good model of an animal cell. In an animal cell, you'd expect to see mitochondria. It's a eukaryotic cell, so we know we'll have a nucleus with chromatin in it. We'll have our Golgi apparatus, which is part of the endomembrane system. There should be mitochondria. Oops, sorry, got ahead of myself. Good. And smooth and rough endoplasmic reticulum. Here's a model of a plant cell. Here's that large central vacuole I mentioned. And there's some chloroplasts as well. Not all plant cells have chloroplasts, but many do. There's also mitochondria, skeleton, the nucleus with the nuclear envelope, and the nucleolus. Good. And notice the plant cells also have mitochondria. Oh, and there's the ribosomes. Good. All right, mitochondria. This is the location where cellular respiration happens. Mitochondria have a double membrane. There's an outer and an inner membrane. The inner membrane is folded up upon itself to increase the surface area. The cristae are where enzymes are stored for ATP production, and it increases the surface area to increase the amount of ATP made. Matrix is the fluid-filled inner compartment. All right, chloroplasts. These are the site of photosynthesis. There's a double membrane. They have thylakoid discs in stacks called grana. So one of these little peppermint patty looking discs is a thylakoid. A stack of thylakoids, like this, a stack of thylakoids is called a grana. And the fluid that fills in the voids within a chloroplast is called stroma. The thylakoid membranes are rich in chlorophyll pigments. These are the pigments that capture sunlight energy. All right, and something I think is super cool about mitochondria and chloroplast is this. We call it the endosymbiont theory. I think it was the 1980s, a biologist by the name of Lynn Margulies came up with this really elegant explanation. Mitochondria and chloroplasts are unique among cellular structures in that they have two layers of membrane. There's an outer membrane and an inner membrane. And Lynn Margulies was wondering, well, how in the world could that happen? Why don't all the other membrane-bound organelles have two membranes? And this is what she came up with. So if we have an ancestor to all eukaryotic cells here, if this ancestor cell engulfs a non-photosynthetic prokaryote, which becomes a mitochondria, like if it goes to eat it, but instead of digesting it, it just lets it live within the cell, then the cell can begin to reap the benefits of having that mitochondria do cell respiration for it. We'll learn more about cell respiration in an upcoming video, but it's the source of energy, or how you get energy from the food you eat. So it's very important. All right, so if a mitochondrion gets engulfed, or more or less eaten, by another larger eukaryotic cell, or pre-eukaryotic cell, if you want to think of it that way, if it doesn't digest the mitochondria and it gets to live, that's a really special symbiotic event, right? And that's why we call it the like the internal symbiont theory. The same thing we believe happened with chloroplasts. There was an ancient photosynthetic prokaryote that was engulfed by a eukaryotic cell, and it was a beneficial relationship for both. The chloroplast gets protection from predators, and the host cell gets the benefit of the glucose sugar produced by photosynthesis. Some evidence to support the endosymbiont theory is that mitochondria and chloroplasts share a similar origin. Prokaryotic cells are engulfed by the ancestors of eukaryotic cells. The evidence for this is that double membrane structure. Like, imagine something that already has one membrane going through the cell membrane of another thing. It would pick up an extra coating of cell membrane. In addition, mitochondria and chloroplasts have ribosomes and DNA of their own, and these reproduce independently within a cell. All right, one more cellular structure I want to talk about is the cytoskeleton. It's a network of proteins and fibers that help give the cell its shape. The functions are support, motility, and regulating biochemical activities. Here's a table to give you some more background information. Cytoskeleton is made up of microtubules, microfilaments, and intermediate filaments. So, the cell wall is a structure that we see in plant cells, and it helps protect the plant and help the plant maintain its shape. It's made of cellulose, which, if you remember, is a carbohydrate. And it also has plasmodesmata, or channels between cells to allow the passage of material from cell to cell. All right, we call this collection of stuff that hangs out in the cellular matrix, but it's just hanging out outside to kind of give everything some structure. Outside the plasma membrane of animal cells, and it's composed of glycoproteins or collagen. The function of the extracellular matrix is that it strengthens tissues and transmits external signals to the cell. All right, tight junctions are when two cells are fused together to form a watertight seal. Desmosomes are like rivets. They fasten to adjacent cells in strong sheets. And lastly, gap junctions. These are channels through which ions, sugar, and small molecules can pass. All right, so some things that only plant cells can boast. They have central vacuoles, chloroplasts, walls of cellulose, and they use plasmodesmata. Animal cells, in contrast, contain lysosomes, centrioles, external structures like flagella and cilia, and the desmosomes tighten and become gap junctions. Animal cells also are the only type to have extracellular matrix. All right, here's one last summary slide. We're talking about the structure of the nuclear membrane. Oops. You can fill this in. The structure of the nuclear membrane is that it's perforated by nuclear pores and nuclear envelope continuous with the endoplasmic reticulum. The function of the nucleus is that it houses and protects the chromatin, which are DNA and proteins, contains nuclei where ribosomal subunits are made, and pores the entry and exit of materials. All right, we also have ribosomes. They're made up of two subunits of ribosomal RNA and proteins, and they can be free in the cytoplasm or bound to the endoplasmic reticulum. Ribosomes are the site or the location of protein synthesis, where proteins get made. All right. The next up here we'll review is the endoplasmic reticulum. It's an extensive network of membrane-bound tubules and sacs, and it's continuous with the nuclear envelope. Golgi apparatus looks like stacks of flattened membranous stacks, and it has polarity. It's got a side that receives, the cis side, and a side that passes on a message called the transphase. A lysosome is a membranous sac of hydrolytic enzymes. And lastly, here we have a vacuole, which is a large membrane-bound vesicle. Most commonly, this would be found in animal cells. Central vacuoles are found in plant cells. All right, the mitochondria here is bounded by a double membrane. It has inner membranes with foldings, typically 2 members around the house, which contain thylakoids stacked up into carina. All right, peroxisomes. These are specialized metabolic compartments bonded by a single membrane. All right, that wraps up our first lecture here on cell types. So by now, you should be able to demonstrate an understanding of basic cell structures and functions, identify the cell types, compare cellular structures between cell types, and describe the functions of those cell structures. As always, if you have any questions, please make sure you jot them down. You can bring them to my student Zoom hours or to class so we can chat about them.